Hi everyone, my name is Farwa Sajadi and I'm a PhD candidate in Dr. Jean-Paul Paluzzi's lab at York University, Toronto, Canada. And today I'm going to be presenting some research examining the roles of ITP and ITP-L neuropeptides in the female mosquito Aedes aegypti. So insect neuropeptides have been a prominent area of study over the past several decades as they play crucial roles in regulating a variety of developmental, physiological, and behavioral functions. These processes include, but are not limited to, homeostasis, water balance, behavior, development, and reproduction. Similar to other animals, the Aedes aegypti mosquito elicit hormonal regulation of peripheral organs through coordination of the nervous and endocrine system. The neuroendocrine system is composed of neurohormones derived from the nervous system, and these signaling molecules include neuropeptides and biogenic amines. Neurons that produce neurohormones are called neurosecretory cells, and in the Aedes mosquito, they can be found in the brain, the subesophageal ganglion, which is fused together with the brain in the adult Aedes aegypti mosquito, in the thoracic ganglia, and in the eight abdominal ganglia, which together with the thoracic ganglia comprises of the ventral nerve cord. These neurosecretory cells project their axons, innervating neurohumoral organs to release these neurosecretory products into the hemolymph. Belonging to the crustacean hyperglycemic hormone superfamily are two peptides. We have the ion transport peptide, ITP, and its alternatively spliced variant, ITP-like or ITP-long, which I'll just refer to as ITP-L. The insect ITP was originally identified in the desert locust, Schizocerca gregaria, where it was shown to facilitate chloride-dependent movement of fluid across the ileum, hence suggesting a role as an antidiuretic hormone. And in 2007, Dye and colleagues identified a conserved ITP gene in the mosquito Aedes aegypti, and this conserved ITP gene in the mosquito encoded for two transcript variants. We have ITP, which is the amidated C-terminus 76 amino acid sequence here, and ITP-L, which is the longer transcript variant at 81 amino acids with an unblocked C-terminus. Similar to the CHH family of peptides, the first 40 amino acids are conserved amongst insect species, with these 6 cysteine residues proposed to participate in disulfide bridge formation. Studies have revealed that both peptides share a common N-terminal sequence, whereas the C-terminal sequences diverge significantly. Thus, these two peptides are predicted to arise from alternative splicing. While studies examining the role of ITP and ITP-L are limited, studies have suggested functions of both peptides as regulators of ion and fluid transport across the ileum of the desert locust, in ecdysis in the Manduca secta, in regulating ovarian maturation in the red flower beetle, and in regulating thirst and excretion and clock neuron modulation in the Drosophila melanogaster fruit fly. However, to date, the role of either ITP or ITP-L has not been elucidated in the Aedes mosquito. So the objective of this study was to characterize and investigate the role of both ITP and ITP-L in the Aedes aegypti mosquito. And to do this, the first step was to investigate ITP and ITP-L expression profiles in the Aedes mosquito. Previous studies in Drosophila have localized ITP-producing cells to the central nervous system of the insect, specifically in the brain and in the abdominal ganglia. As a result, we hypothesized that both ITP and ITP-L expression would be highest in the central nervous system of both adult male and female mosquitoes. So to examine this developmental expression profile for the putative ITP and ITP-L neuropeptides, we collected and isolated RNA from different stages of the mosquito, including the 4th instar larva, early and late stage pupa, and 1 and 4-day-old adult male and female mosquitoes. And we can see here the ITP expression profile and ITP-L. And looking at the developmental expression profile shown relative to the 4th instar larva, we can see that for both ITP and ITP-L, highest expression is found in the 1 and 4-day-old adult male mosquitoes. We then decided to isolate different tissues from 1-day-old adult male and females to examine the spatial expression profile. So we collected the midgut, which is the site for digestion, the malpighian tubules, which is the site for primary urine formation, the hindgut, which acts as the main reabsorptive organ, and the reproductive organs. We also collected the head, thorax, and remaining carcass of the mosquito. So similar to the Drosophila immunos, we saw that ITP was exclusively localized to the head of 1-day-old adult male mosquitoes. And looking at this, we then decided to specifically look at the central nervous system tissues. And here we can see that ITP expression is exclusively found in the brain of the adult mosquitoes. And this is found in both the male and female mosquitoes. Looking at ITP-L expression, we found highest expression levels in the carcass of the male mosquito, which does hold the abdominal ganglia, which we did see highest expression levels. And this was found in both the 1-day-old male and female mosquitoes. So now that we've mapped this expression profile for both peptides, we then wanted to determine the distribution of ITP and ITP-L peptides within the nervous system. And based on our expression profiles, it was hypothesized that ITP would be localized in the brain and ITP-L to the abdominal ganglia of the mosquito. So we were able to get a hold of a primary antiserum specific to the Aedes ITP peptide. And using whole mount immunohistochemistry, the central nervous system from 1-day-old male and female mosquitoes revealed ITP-like immunostaining in these 3 pairs of lateral neurosecretory cells in this medial region of the brain hemisphere, along with a single pair of neuropeptides along the anterior region of each brain hemisphere. And we can also see these projections leading to the anterior portion of the brain over here. ITP-like immunostaining was also observed in 1 pair of neurosecretory cells located medial-posteriorly on each abdominal ganglia. So abdominal ganglias 1 through 7. And this image over here is representative of the 2nd abdominal ganglia. So now that we've determined the distribution of both ITP and ITP-L within the nervous system, the next step was to elucidate any putative physiological roles. So as previously mentioned, ITP has been shown to act as an antidiuretic hormone in the hindgut of the locust, and also been shown to regulate thirst and excretion in the Drosophila fruit fly. As a result, we wanted to investigate any putative role in feeding or ionoregulation in the adult mosquito. So initial attempts in examining a putative role of ITP and ITP-L was examining its role in feeding and starvation. So adult mosquitoes were isolated post-emergence and placed into 3 treatment conditions. We had a fed treatment where mosquitoes had access to a sugar meal, a non-fed treatment where the animals were deprived of both food and water, and a water-fed treatment where they had access to water but no sugar meal. And these adults were collected after 24 or 48 hours, and mRNA transcript levels of both ITP and ITP-L were measured. So if we look at the 24-hour treatment first, we can see no significant changes in ITP transcript levels for either treatment. However, in 48 hours, we see a significant enrichment of ITP transcript levels in the non-fed treatment in both male and female mosquitoes. Similar results were seen with ITP-L, where no differences were seen in the 24-hour treatment. However, after 48 hours, we do see this upregulation of ITP-L mRNA levels in these starved non-fed male and female mosquitoes. An enrichment of ITP and ITP-L transcript levels in this 48-hour non-fed condition could potentially suggest an antidiuretic role, since ITP and ITP-L could play a role in preventing desiccation in the adult mosquitoes. Since we noted a potential role in feeding and starvation, we next wanted to examine whether a protein-rich meal influences the transcript abundance of ITP and ITP-L peptides into the adult female hemolymph. So to do this, we introduced a blood meal to females and collected them after 1, 6, 12, and 24 hours post blood meal to examine a time course response of ITP and ITP-L transcript levels. Blood-fed females were then compared to a control sucrose-fed female. So here we have the results for ITP transcript levels, and we can see that at all time points, there's no significant changes in transcript levels post blood meal. However, when examining ITP-L, we see a significant upregulation of transcript levels 6 hours post blood meal, with levels slowly dropping at 12 and 24 hours post blood feeding. So just to summarize the few key points that we've seen so far. So through our expression analysis profiles, we see that ITP expression is exclusively found in the brain, and ITP-L expression is found predominantly in the abdominal ganglia. ITP-like immunoreactivity was found in 4 pairs of neurosecretory cells in the brain, and ITP-L to 1 to 3 neurosecretory cells in each abdominal ganglia. And through our bioassays, we're potentially suggesting a role of ITP and ITP-L in starvation and blood feeding. And gaining a more comprehensive understanding of these neuropeptides and signaling systems can allow us to further unravel this complex neuropeptidergic regulation in the Aedes aegypti mosquito to ultimately enable more efficient strategies for disease control against these important disease vectors. Thank you so much to my lab and to Beyond Science Initiative for allowing me to present my research. Thank you so much.